Good morning and good afternoon, whatever friends. So let me first give some introduction while we are waiting for some more friends to join. So first of all, let me welcome everyone to today's talk. The title is Innovative Solutions for Plant Breeding, a biotechnological research company perspective. So the speaker is from a biotech company. It could be quite different from the academics. This is the 6th talk of a series targeting to promote agrobiotechnology and enhance understanding of the potential applications of agricultural products. We hope that the series will inspire international scholars, researchers, farmers, and business in the field, in the agricultural field, as well as the interested public. First, let me introduce our speaker, Dr. Michiel van Eijk from the Netherlands. He obtained his Master of Science in Animal Sciences from Lenin Agricultural University and the PhD in Animal Sciences from the University of Illinois at Urbana-Champaign. He performed postdoctoral research as a molecular biologist at the Netherlands Institute for Developmental Biology and as a molecular geneticist in the Wilhelmina Children's Hospital. So in 1997, Michiel joined T-Gene and served in various R&D and IP manager roles. In 2006, he completed Foundations of Management at the Yanmar Business University, and in year 2009, the High Performance People Skills for Leaders programs at London Business School. Between 2012 and March 2020, he was the Chief Scientific Officer of T-Gene. In 2018, Michiel joined the board of Holland Bio, the Dutch biotech industry association. Since March 2020, Michiel is the Chief Executive Officer at Genetwister Technologies. Additionally, he is the author of more than 40 peer-reviewed papers and co-inventor over 30 patent applications, mostly in the field of genotyping technology development and the next generation sequencing application. In the following presentation, he is going to provide you an overview of research projects performed in a contract research company owned by several multinational vegetable and horticulture plant breeding companies, and so how such research has been organized. Now let's welcome Dr. Van Eijk to the floor. Michiel, please.
Well, thank you very much, Ming, for this extensive introduction and for inviting me to participate in your seminar series. I'm honored to be able to contribute to that, and I would also like to thank Joanna for her help in the preparation of this seminar. Let me try to share my screen. Can you see my first slide?
Yes, we can see.
Yeah. Okay. All right. So, well, as you indicated, the title of my presentation today is Innovative Solutions for Plant Breeding, Biotechnology Research Company's Perspective. And what I had in mind today for the audience is to share with you some insight into, well, the research topics of interest to vegetable and horticultural breeding companies, obviously, but also, how research is actually organized in a contract research company, which is in some aspects possibly different from in an academic setting. Well, the outline of my presentation is I will say a few words about the history of Genetwister Technologies, the unique model behind the company in which seed breeding companies work together in R&D, even though they are competitors in the market. I will share with you some example research and development projects. say a few words about our track records and results that we have achieved and aim to achieve. And of course, there will be room for questions that you may have. So to start off with the mission of the company, it is Genetwister's mission to develop and make the best use of innovative methods, techniques, and plant genetic resources to create value for our clients' breeding programs. There's a lot of words in this sentence, but they are carefully thought about, of course. But what I would like to point out is that it is very important for us as a company that we create value, so that we do things that are appreciated and translate into business opportunities for our clients. So we have a focus on the needs of our clients, logically, and our clients in this case are also our shareholders. They are seed companies whose business is to develop new varieties with improved traits and sell either the seeds or other plant products to the growers, and eventually they produce the fruits and the vegetables that we eat, and the flowers that we have in our gardens or in our houses. So a little bit about the history of the company. Genetwister was founded in 1998, so more than 20 years ago already, by two entrepreneurial scientists of the university here in the town of Wageningen, the Netherlands, where we are based. Wageningen is a small town with about 30,000 people, of which the vast majority are students who are attending the agricultural university, the only agricultural university that our small country has. And the founder started this company at that point in time because genetic modification technologies were at a very early stage. And in order to do that, tissue culture technology is very important in order to make use of the opportunities provided at the time of genetic modification. So the insertion of foreign genes to affect certain traits of a plant. And for the first decade of Genetwister's history, it was a very small company with about 7 people. And in order to further grow the company, in 2009, two seed companies called Bio Seeds, which is a company in the Netherlands, and East-West Seeds, which is a company headquartered in Thailand, both vegetable seed breeding companies, became shareholders of Genetwister, and Genetwister started to do research for these companies as their clients and shareholders. And so this was the start of essentially a consortium of companies who are both acting in the same commercial market, but nevertheless decided to work together and perform part of their research with their daughter company, Genetwister. Well, this consortium has grown over the past decade. In 2011, another Dutch company called Dumen Orange joined the consortium. Dumen Orange is a horticultural company, so they only work with flower species. In 2013, a seed company based in Taiwan called No Seeds joined the consortium. And in 2014, a multinational company, Sakata, headquartered in Japan, joined the consortium. So since that time, Genetwister has 5 shareholder companies active in vegetables or horticultural species, and we form an alliance with them, a consortium of companies working together in R&D. So why would companies who compete in the same market work together? That's rather, let's say, uncommon, you may think. But there are really certain advantages of doing that. So the advantages of working together via a daughter company, in this case Genetwister, is that what is called pre-competitive research, so research that has a high risk of failure, but of course a high value if successful, is costly and risky, and it may be in the interest of all to share that risk and work together. So there is the shared exploration of new research opportunities emerging from academia and also from commercial entities. And what we do, or part of what we do, is to scout continuously for such opportunities. And in that sense, Genetwister acts as the first filtering step to identify technologies, interesting organizations to collaborate with. I already mentioned another advantage is that investment risks and benefits can be shared if you work together and share the costs. Another advantage is that you generate an increased critical mass in R&D if 5 companies work together. Together, you know more than alone, we say in Holland. So we are also serving at Genetwister as a discussion platform of peers for the scientists in these 5 companies, which provides them with the opportunity to benchmark their own choices. Well, we are based in Wageningen. We have a laboratory and, well, we have expertise, of course, with the people that work for our company. And the vision behind that is that this provides complementary resources to the internal R&D performed by each of these companies. So we are a part of their R&D efforts. Well, if we are successful, of course, that leads to proven technologies that can be implemented either in Genetwister itself to provide subsequent projects and generate the results for our clients, or sometimes we transfer technologies and tools towards these breeding companies, and that's called the technology transfer mechanism. So the scientists in our company frequently visit our shareholder companies to discuss projects, discuss results, transfer tools, and help them expand their own internal infrastructure. Well, the final 2 advantages are that there are possibilities sometimes to establish IP, so intellectual property to protect certain valuable technologies or results. And by having a daughter company, the value of that company itself may create shareholder value. So there's multiple reasons why working together as companies acting in the same commercial space can be very attractive. Well, what do we actually do? What is our innovation strategy? What are the areas of interest where Genetwister works? Well, there are 4 main categories. Very important is all the opportunities related to next generation sequencing, which is driving our genotyping methodologies, haplotyping methods, the creation of reference genomes, nowadays pan-genomes of various diploid and polyploid plants. You can imagine that working with vegetable companies and with flower companies, I think together they sell seeds or plants of more than 200 species. So there's plenty of work for us to do. Well, in order to structure all that information, it's very important to store information in a structured way and provide tools to access sequence data and actually derive useful information from that. Data is one thing, but more important is to extract information or generate valuable information from the wealth of data that can be generated nowadays with, for example, next generation sequencing equipment. With all these tools and sequences and knowledge at hand, ultimately, for breeding companies, it's about the development of new varieties with improved traits. A unique and important, I should say, area of expertise of Genetwister is the area called genomic selection and genomic prediction. So these are methodologies to improve particularly on complex traits. And later on in my presentation, I will illustrate that in more detail. And related to that is the methodology to extract phenotypes. Ultimately, breeding is often about, or exploitation of variation is about linking genomic variation to the phenotypes. And of course, the next generation sequencing drives all the capabilities on the genomic side, but equally important is the scale and the accuracy of generating phenotypes, and there are a lot of developments in the area there as well. that are very interesting to work with also scientifically. And the fourth component of our innovation strategy is the lead discovery. All these methodologies should lead to identification of genes that underlie these traits, such that beneficial alleles of these genes that occur in nature can be selected to contribute to the improved varieties, or these genes can be used as a target for example, mutagenesis technologies or genome editing technologies. So essentially to introduce new variation in these genes. But for that to be possible, you need to know which genes to target. So these are the main elements of our innovation strategy. Well, how do we organize this? How do we structure this? And what is essentially our business strategy? Well, our business strategy as a company is to structure all activities in the form of projects. And what that means is that before we engage on a research challenge, we define what are the goals precisely, what are the materials that we need for that. So everything, well, that's familiar, of course, also to academia, where grant proposals are prepared. But in a fair amount of detail, we describe what we aim to do, and also in which time frame and with which budget. We have 4 teams in-house: a genomics team, a bioinformatics team, a genetics and breeding team, and a software development team. And essentially the projects that we do incorporate expertises from all these 4 groups, depending on, of course, the need for particular expertise. But it's very important that there is internal collaboration, flexibility, and teamwork, which is driving success, of course, in any organization. Well, in terms of the type of projects, it's interesting, I think, to point out the various types of projects, given the fact that we work with companies who are also each other's competitors. So first of all, it is important that there are very clear agreements about which information is shared with which company. Of course, if company A pays for a project, the results should not be accessible to company B if they are not part of the project. On the other hand, there's a reason for working together in a consortium. So on main lines, the projects are organized along 2 lines. First of all, what you see at the top of this slide, there is a joint multi-year strategic R&D program with all the shareholder companies. So this is essentially the reason for them to work together. This multi-year strategic R&D program, in which all partners are equal, aims at, for example, developing tools that are of use for all of them. And this is essentially, this is actually the reason why it is attractive to work together. But once these tools are there, we also do projects which we call bilateral projects with each of the companies separately to generate interesting results, for example, for a particular flower or vegetable crop of interest, of which the company wants to improve a certain trait and create a new variety that eventually will be sold in the market. So these are projects that are done with one of the companies, and of course the results of those projects will not be shared with the others. And this model is, well, has proven itself over the course of many years. The multi-year program, of course, provides tools and technologies mainly, and the bilateral projects provide what we call results that have a shorter term use to be incorporated in the breeding process of the seed companies. Well, now some examples what these projects can be. A very important area I already mentioned when I touched upon the next generation sequencing space is de novo genome assemblies. Why are genome assemblies so important? Well, a very interesting paper in that respect is the paper that was published in Giga Science by Benevenuto, who analyzed the impact of working with high quality haplotype resolved genomes of a blueberry. And for that, they used a high quality haplotype resolved reference genome of a variety called Draper, which was actually generated with a combination of techniques. It was based on Illumina sequencing, supplemented with 10x chromium sequencing, and ultimately high C scaffolding. And these results were compared with a draft genome that had been generated earlier on another variety called W8520. So that's on the left-hand top of this figure here. That was generated with older technology, 454 pyrosequencing and Illumina, so only short read sequences. And the result of these genome assemblies was that the older technology resulted in 13,700 scaffolds, whereas the high quality genome was completely chromosome and haplotype resolved. And these researchers investigated or were asking the question, well, how would a high quality reference genome affect the previous results with the lower quality genome and future research? And they looked at the ability to unravel the genetic architecture of certain traits in blueberry, the ability to mine candidate genes, and also to do genomic prediction. And a very interesting observation was that using the 8520 reference, so the lower quality genome, when investigating markers associated with a trait, the amount of a certain volatile called geranyl acetone, you see all these red dots here of associated SNPs, which were actually scattered throughout this abundance of 13,000 unplaced scaffolds. And that led to the impression that this was actually a multi-genic trait in which many gene regions are involved. But if you look, when they reanalyzed that SNP data with the high quality reference genome, and that's in the right side panel, it became clear that all these associations were actually in the same region of this so-called Manhattan plot. So this tower of the Manhattan plot emerged. And instead of the suggestion that this was a polygenic trait, it became clear that most likely this trait is actually controlled by two loci, which have a major influence on the trait variation. So this makes clear in this picture alone the importance of working with the highest possible genome sequences in order to perform trait improvement. So that's something that we are very much aware of, and for that reason generating such reference genomes is what we do very often for our shareholder companies or other clients for that matter. And this is an example of a chromosome level bitter cord assembly that we completed actually a year and a half ago using PacBio sequencing. So that's what is called long read sequencing in combination with bio-nano optical mapping. And bitter cord is actually a rather small genome, 339 megabase pairs, and it is diploid, so it's in that sense not the most difficult one. But the point is that we try to leverage the capabilities of current next generation sequencing technologies in order to provide these high quality reference genomes to our shareholders. And these are some statistics of this bitter cord genome, and 50 of 12 MB, so that these are very respectable sizes of contiguous genome sequences. So and in fact 99% of this PacBio assembly was contained within the 11 chromosomes, as one would expect from a high quality reference genome. And when put in perspective on this slide, where you see on the x-axis the N50 contact size, and on the y-axis the genome size, then bitter cord ends up somewhere here. So from all the sequences, sequenced genomes that are plotted in this graph, based on the various sequencing technologies that have been used over the past decade, I would say, to create these reference genomes. using PacBio technology puts it at the desired end of the spectrum, so to say. Now switching topics to another important area of research in our company that relates to genotyping technologies. That's an area that I'm personally very familiar with, as I spent a sizable portion of my career on the development of genotyping technologies. You probably know that this is a very dynamic field where different workflows are generated by companies and academic research groups all the time. And that's a very good thing, of course. And it also means that scouting is an activity that we need to do continuously, because there are always people who come up with smarter and better ideas to drive down the cost per genotype, and that's very important for us. So this is just a snapshot overview of publications and names of technologies that are either genotyping SNPs in a targeted fashion or in a random fashion or combinations thereof. But for breeding companies nowadays, genotyping technologies are very important, and especially because genotyping can still be a very costly, let's say, a part of the development of new varieties. Because of the large number of samples that are processed in a company per year. So that's why genotyping technology development is part of this R&D program to provide our shareholders with the most cost-effective means to genotype the species that they are interested in. And for example, in the case of ornamentals, this is an example project. Many ornamental species like tulips, etc., are polyploid. Sometimes they have 6 or 8 genomes, and that makes genotyping rather complex, or at least it makes it more difficult to provide accurate genotypes, which of course is what we're interested in. And this is an example of a project where we generated haplotypes in a polyploid species using hybridization capture, target enrichment, followed by sequencing with Illumina short read sequencing, paired and 300 base pair reads, and then try to resolve the haplotypes using appropriate software tools for that. And the output of that is then that you can see the various haplotypes for certain regions of that were targeted by the hybridization capture probes. And these are important, let's say, results for in this case, the company who is breeding horticultural species. Well, how do we cope with all the data that are generated? I mentioned reference genomes. I mentioned genotypes. I mentioned the large number of species that we work with. So in order to make optimal use of all this information, it needs to be stored and structured and accessible for our clients. And for that, we have built a platform that we call CropAware. It's a database infrastructure coupled with tools to access these data and tools to analyze data. And this is organized in such a way that it provides project-based access to navigate crop genomes. So it's organized per crop. All the information we have either generated ourselves or from the public domain is incorporated in this database. And we provide our clients access via a secure way, of course, and then they can analyze the information or search for genes or perform blast analysis or look for sequences observed in one crop, whether they have autologous sequences in the other. So various, let's say, research questions that are important in order to understand the genetic variation of their crop of interest. And that's the reason why, as part of our team is dedicated to software development. Well, I mentioned before genomic prediction and genomic selection as one of the expertise areas. To put a little bit more context around that, in traits we make the distinction typically between monogenic traits, so these are traits that are essentially controlled by a single gene. Oligogenic traits, those are traits controlled by a handful of genes, but not that many. And then the real challenge to improve is what are called the complex traits. And those are traits that are controlled by many different regions in the genomes, and for which then potentially many different SNPs can be identified, which together may be useful to improve such a trait. And well, you can imagine that unraveling the complexity of finding markers for a monogenic trait is relatively straightforward with the tools that are nowadays out there. But for complex traits, that's a different ballgame. So we make use of a very, let's say, powerful way to address the challenges provided by complex traits by using genomic prediction and genomic selection. And the way this works is as follows. It starts with a population of what is called a reference population, which is phenotyped for the traits of interest, and also genotyped. And this reference population is essentially used as a training set to find out in this set of samples which markers are associated with the phenotypes of interest. And that's actually then used, that information, to build a prediction model whereby the contribution of each individual SNP that is measured in this reference set is calculated. How much does this contribute to the trait? And of course that depends on, among others, the linkage to the trait, linkage disequilibrium, genetic structure in the population, etc. But the prediction model is basically the model that measures the combined effects of all the SNPs, the genotypes that have been used in the reference set. And that information, that model, is then used to predict for the selection candidates what the phenotype will be if they are only genotyped. So in the selection candidates, the phenotyping is not performed, but only the genotypes are measured. And based on the model, then a prediction is made. What will the phenotypes of these lines be? And the parents for breeding are then selected based on that information. And that approach has several advantages, because phenotyping can be very costly. Sometimes phenotypes can only be measured at a later stage of development. And by being able to predict the phenotype using genotypic information, the breeding process can be accelerated, essentially. So there is a reference set and a selection of candidates, and then the prediction of the parents. That's the essence of genomic prediction and genomic selection. This is especially powerful for unraveling complex traits where, many genes are contributing a very tiny portion of the variation of this trait. What we do is leverage these types of technologies to help breeders develop varieties faster. So for monogenic or oligogenic traits, that means the one or few regions involved have a large effect. Few regions explain a large proportion of the variation, and that means that we only have to check those genotypes to predict the phenotype with a fairly high accuracy. For polygenic traits, as I mentioned, there are many regions involved with small effect sizes, and we need to check genome-wide the genotypes of many markers to predict the phenotype, and this is called the genomic prediction. Well, some examples again. This was a project that we did with our shareholder Beozaden in the Netherlands, who was interested in finding a marker for a trait in lettuce, a trait that was expected to be monogenic. And what you can see here on this slide is that indeed we found a SNP marker where the homozygous A allele was associated strongly with the lowest phenotypic scores, and the homozygous B genotype was associated with the highest phenotypic scores. And if you look at, let's say, the genome-wide association, that's also depicted here, you see that there is one area in the genome where multiple linked SNPs basically cluster together. And this is probably where a gene lies that explains the large proportion, if not all of the variation, or nearly all of the variation in this monogenic trait. These are straightforward cases to unravel, although as long as the tools are in place to do that, in this case, a 90,000 marker SNP array, and that was used for to genotype 576 accessions. Well, this is an example of another project with two other shareholders, East West and Novo, who did this project together. A genome-wide association analysis of polygenic traits, and what you then see immediately, if you look at the right-hand side, there is not such a clear-cut association between the genotypes of individual SNPs and the phenotypic measurements, although there is some tendency. You see three A homozygous genotypes in the lowest phenotypic score class, and you see a slight excess on this limited, illustrative graph of the homozygous G allele. But if you look at the Manhattan plot, you see that there are multiple regions where markers lie that seem to be associated with this complex trait. So yeah, this is where the power of this genomic selection and genomic prediction technology is, of course, more relevant. Another project that we did with one of the shareholders was, well, it's nice, these new techniques, but how well do they compare to the traditional selection? So this was a project in pepper where trait improvement was done via either the conventional selection or the genomic selection, and then the results were compared side by side to see, okay, what did we learn from this? What does this mean about the real added value of using genomic selection for, in this case, certain traits in pepper? And these types of, let's say, research questions are answered and they determine or their impact on how seed companies adopt new technologies to become part of their breeding efforts. And in this case, well, there were certain advantages noted related to genomic selection. For example, well, the process was a little faster. So, and that was because, well, certain selections could take place off season because you only need the marker information. You do not have to complete the full season in order to obtain the phenotypic information. That translates into faster cultivar development. Genomic selection is also used often to select for multiple traits simultaneously. And I will come back to that later when I show some of the tools for that. And it's mostly applicable for selection of traits with a low heritability. And it also has the advantage that it maintains genetic variation maybe better because by using information on the combined effect of genetic markers, one avoids the situation that certain markers are becoming fixed in the population in a relatively short period of time, which would reduce the genetic variation in the germplasm too quickly. Yeah, I just mentioned the tools. So this is just a snapshot to illustrate what we do in terms of tool development. So for genomic selection, we have built our own tools to visualize experimental data and run certain analyses on the data sets that we generate. The data from multiple traits can be combined in a selection index, and the relative weight of each of these traits in the combination can then be influenced by the researchers by basically defining which percentage a certain trait may represent in the index. And at the end, of course, all these analyses need to be reported in a certain fashion, and you know, these tables should be exported to Excel or whatever is necessary to have a detailed look at that. So yeah, maybe to summarize on the, let's say, the content level, what Genetwister has done over recent years is to build a number of de novo whole genome assemblies. I showed the Bittercord example. Another one was, I did not present in detail, was an assembly of rows, and this was actually the first haplotype resolved genome assembly of a polyploid genome. We have generated and are generating many first reference genomes of commercial vegetable crops and ornamental species. We are now transitioning towards pan-genomes. So because of the lower cost of sequencing that's still going down, you know, that means that more genomes can be sequenced for the same price, and that it now becomes feasible to generate multiple reference genomes per species. And that's very important because a considerable fraction of genetic diversity cannot be identified by having a single reference genome and then only performing short read sequencing and map that to the single reference genome. So ultimately, of course, all varieties would be de novo reference genomes sequenced, but that's way too costly. But pan-genomes is a step in that direction. Well, we established such a pan-genome for 17 tomato lines as an example. I briefly described the crop-aware infrastructure, the tailor-made software for genome sequence data analysis, integration, and visualization. And well, I just illustrated what we do in terms of genomic selection and genomic prediction in crops. And well, we are actually the first to apply that in a commercial breeding program in vegetables for our shareholders, and in ornamentals in chrysanthemum. And I just showed a tool that we actually have transferred to our shareholders to do these type of projects now themselves in-house. Maybe to conclude this presentation, what is our vision for the future? What are our points of, let's say, of interest? Well, the cost of next generation sequencing is still going down. I think that will lead to a gradual replacement of genotyping methods or targeted genotyping methods by whole genome sequencing. In some species with small genomes, we have already made that transition. And what that means is that in such species, we do not use, let's say, a genotyping array with 90,000 SNPs, as I showed for lettuce, anymore, but we just sequence the whole genome, or a population of genomes. If we have a project with 300 lines, we decide to sequence all 300 instead of genotyping these 300 lines. And I think with decreasing cost, that transition will become feasible also for larger genomes in the future. I just mentioned the shift from a single to a few reference genomes and ultimately towards pan-genomes. I didn't say too much about that yet, but digital phenotyping is complementing what is called the breeder's eye. Many traits in breeding companies are selected based on the expertise and of breeders who have extensive knowledge of their crops, and yeah, it's almost an art to do accurate phenotyping. But in order to scale that to larger volumes, or to measure things that the breeder's eye simply cannot see, digital phenotyping is an area of interest in crop improvement that is developing heavily, translating into larger data sets, objective measurements, higher accuracy, and cost reduction. So it's very important for breeding companies. In that context, phenomic selection is an area we are very much interested in. And yeah, this concept was described in a recent paper by Vincent from France et al. In 2018. And what it actually does is to use near infrared to measure the composition of certain plants, parts, for example, seeds or leaves, or well, maybe fruits, and use that as an improved predictor for complex traits. So instead of using SNP genotypes to build the model for genomic selection, it's actually measuring composition aspects to build the model. And by measuring the composition, it it essentially also integrates, let's say, effects that are due to interaction effects in the genome, which you do not measure if you measure SNPs individually. So I think it's a fascinating area that's at the early stages still, but potentially very promising. And it also illustrates that, you know, there's a lot of, let's say, other omics layers in between the genotypes at one end of the spectrum, or the genome at one end of the spectrum, and the phenotypes at the other end of the spectrum. And in order to leverage all that knowledge and expertise, data analytics and lead discovery are becoming even more important to improve crops. So yeah, well, with that, I hope that I have provided you with some insight into well, who we are and what we do, and why in a commercial environment companies work together. So with that, I thank you for your attention. I'm happy to answer any questions you may have.
Thank you, Matthew. So before we go to the Q&A session, as a tradition, we talk pictures. So could everyone turn on your video for a while so that we can take a group photo together? So who's taking it? Joanna, you're taking the photo, or?
Yeah,
Okay.
I'll take the photo.
Okay.
Okay, everyone.
Okay.
Are you ready? So 1, 2, 3. Okay, we take several more. 1, 2, 3. Okay. I still see someone opening the camera. Okay, one more. 1, 2, 3.
Okay.
Okay.
Thank you.
Thank you very much.
Okay, so it's now the Q&A time. So it's very great to have a biotech company chief scientist to discuss with us the strategies. So who would like to ask questions?
So I want to shoot out like 2 questions in 2 different areas. One is that I can see that there are many companies working with your company. So my first question is about all these open database and open forums so that everybody can share all the common goods. I see many new trends, like when you mentioned about NIR, and I also noticed that you actually filed 30 patents. So of course, like every company want return, right? ROI is very important. So I want your insight in terms of this so-called copyleft and also this open source in this particular area. So that's my first area. The second area is of particular interest to me in the space of botanical, which is some degree of functional aspect or therapeutic. I see that you have fugua, the bitter gout as stuff. And so in the, I studied TCM years ago and whatever traditional Chinese medicine. So to me, plants like lavender really help you to soothe and or whatsoever. I noticed that you mentioned about at least 10 times about the ornamental aspect, like the flowers, like whatever. I went to Netherlands, it's just amazing to me. And then seeing the tulips, I have never seen so many tulips in my life, right? But that granted, but then at the same time, I said, how would that your gene mapping together with certain degree of functional aspect in terms of therapeutic movement in your particular field, and is there any, like in that particular space, is there other people start exploring it? I know that Ming is very into crops. I know that saving life, having a full stomach is very important. But then at the same time, I think that in this particular era, if you have some anti-COVID plan, I would definitely plan my whole office with those anti-COVID plants, right? So these are the two separate areas, but it's of a particular curiosity of me, and then by really like reading your PPT and so on and so forth. And so I know this big area, but I'll throw this in the floor and make your pick which one to answer first.
Yeah, I think well, let's start with the first one. Indeed, you know, I think you're asking me how we view open databases and how this relates to, for example, patenting and etc. Well, first of all, you know, indeed, the 30 patents that Ming mentioned, I'm indeed an inventor of many patents, but that was in my previous job at another company, which is, oh, by the way, acting in the same space. But what I can say is,
I'm a patent attorney. So I'm a patent attorney. So when Harman mentioned about 30 patents, I just like, down, my eyes just open.
Yeah. No, but indeed, well, I've been managing a patent portfolio for many years, also as an inventor. But to link it to this topic, indeed, in this company, you know, our goal is to, as I illustrated, to develop tools and technologies at the forefront of innovation on the one hand, and of course use these technologies to create value for the clients in terms of improved traits. And so that latter part is of course more important for them than the technology development per se and the patenting thereof. But it is, I think, my vision that these things go hand in hand. I did not mention that explicitly in the beginning, but our company is only 32 people. So if with a relatively small company, you know, you want to add value for 5 shareholder companies, of which most of them are large multinational companies who have their own internal resources, it is an absolute requirement to think about how you can be unique and how you have something that is really of literally added value to them. Because if it is only a commodity, they can get it from many other parties. So it is a strategic choice to act at what we call the forefront of innovation, so to explore new opportunities. And if that creates valuable new techniques, then it would not be wise to consider or to not patent that. And indeed, I have spent the main part of my career doing exactly that. For some people, it may be a different, let's say, expertise area. But if you understand the connection, which obviously you do, between IP and R&D, then value creation becomes a much more, let's say, motivated activity. And it is from that perspective that we keep an open eye to patents. But to come back to the first part of your question about how that relates to, for example, open databases and open innovation, I think at the same time, and that's what I also meant with the scouting, it is very important to collaborate. Well, actually, I have done that all the time. That's how I met Ming, and we did collaborate. It was part of that vision. You know, things are moving so fast nowadays that if you cannot collaborate, it's very difficult to be successful. And by the way, collaboration is fun. It's nice to work with people from different backgrounds and different cultures, at least I think. Open innovation and working together with academia is very important. With respect to databases, of course, we use open accessible databases, but often for breeding companies, that is not enough because they need to, let's say, structure the information in a way that fits with their own business. And, you know, in the, let's say, the outside world, there's a lot of heterogeneity in the way how information for tomato is stored, for example, compared to cucumber, compared to the brassicas. So in a commercial environment, seed companies often choose to develop a infrastructure which is the same for every crop. That makes it more efficient to work with. So we are kind of that middle man, so to say, that uses openly accessible information, but also develop tools like CropAware to make it more uniform and easily accessible, and also to protect confidential information that the companies are not willing to share with the outside world. So yeah, it's a long answer to, I think, your first question. With respect to the second one, the botanical aspects and the functional elements relating to all the horticultural crops, I think the horticultural crops are even more challenging than the vegetables and the field crops because of their large genomes. Hexaploid or octoploid genomes are a nightmare to work with from a genetic point of view. There's many different flower species, very beautiful indeed, but also the business is completely different. You know, the margins are sometimes very small, so the resources are not there to justify large investments in certain of these flowers. So all of these, let's say, preconditions force innovation. We have to think about smart ways to create value with sometimes limited resources. And that's very early days there. You know, also the horticultural species are of course important plant species, especially indeed in the Netherlands from an economic perspective. And many technologies and tools are equally applicable in vegetables and field crops and in flowers. And that again underpins why it makes sense for companies in this case who are in slightly different areas to still work together.
Okay, so the next question is, I think Dico would like to ask a question, right? So Dico, can you just unmute yourself and ask?
Thank you, Ming. So, Michael, thank you very much for a very inspiring talk. So, unfortunately, yeah, you can't see me because my camera is not working, but I'm relieved that Ming said I shouldn't worry about the camera. So thank you. With regards to the two versions of the sequencing aspects that you talked about earlier on, so we've got the draft genomes, and then we also have the much more accurate, detailed genome sequencing. Sometimes, of course, this is driven by cost considerations. Would the draft genomes be improved if they are complemented by small, detailed RNA-seq exercises, especially if one is looking for differences in expression of certain for the breeding exercise that one is looking at? So that's my first question. And then in consideration of what you showed in terms of the SNPs and the gene calling between the draft genome and the more accurate sequencing, much more accurate genome sequencing efforts. Those two, in the more precise genome sequences that you showed, were they picked up as well in the draft genome sequencing? Well, I mean, irrespective of the fact that there were more SNPs that were not associated with the functioning of the, well, with the phenotypes that were targeted in the draft genome. Did you find that the draft genome that generated more SNPs that were not relevant also resulted in the same two loci, not SNPs, two loci that you picked up in the accurate genome sequencing? Then the last question, I know I'm stressing this, but the last question would be, much of what you have talked about is looking at horticultural crops. And of course, I understand why, because you said so. And of technologies are shared with field crops, of course, as you said. I would just be curious from a partnership point of view now, would Genetwister be interested in genome sequencing for a field crop that might be seen as extremely important in the African environment? And I'm saying this because besides working with soybean and maize, which are important in South Africa, I have started some really interesting work with a relative of cowpea, which is Vigna ingiculatus, that's cowpea. It's got a wild relative that is indigenous to Africa, but it's, in my opinion, much more valuable than cowpea because except for the stems and the roots, one can use every part of the plant. The leaves can be used as a vegetable in the same way as we do with Swiss chard or spinach. And the seeds, of course, can be used in the same way as we use soybean seeds and common bean seeds, while it is one of the very few legumes that form a tuber. But that tuber is high in starch and carbohydrates in the same way as you would find in potatoes and cassava, but it is one of the very few that has a protein content as high as common bean, in the sense that it can contain anything between 15 and 20% protein in the tuber. So I have specific interest in this particular African indigenous crop, of course. So those are my three questions, i.e., would Genetwister be interested in teaming up with us in producing a high quality genome sequence of this other plant that I talked about? But that's in addition to the other questions I've asked.
Yeah, okay. Well, thank you for your question. I think I can answer them in the order that you asked them. Your first question was, well, would an RNA data set be used in assembly? I think the answer definitely is yes, because if you have RNA-seq data, that's very helpful in the analysis of the completeness of the genome. We typically also, if it is available, use that to assess the quality of a genome assembly. Which fraction of the known genes from the species do we actually find back in the genome? So yeah, the short answer is it's very valuable to have an RNA-seq data set. It does, however, not improve the assembly itself, per se. It's more valuable in the quality assessment and the quality assessment of the assembled genome. I hope that makes sense. As to the second, sorry.
I was just saying, thank you. It makes sense. Yes.
Okay. As to your second question, the analysis of the blueberry data and whether, there was a difference between the draft genome and the accurate genome with respect to the SNPs that were not so relevant. That I cannot answer because I have not studied that data set in that detail. But I think the message of the paper is that, of course, the quality of the genome, first of all, impacts on the probe design, which may lead to, the SNPs that are being called in the first place, whether they are there or not. But in terms of, let's say, the association power, I do not think there is that much difference. But the main effect is on the understanding of the complexity of the trait and the number of regions involved, as this Manhattan plot clearly illustrated. So I think it's more in the understanding of the genetic architecture that the difference lies than in the, let's say, the number or the accuracy of the association. And then the third question, would we be interested in teaming up to develop a reference genome of a legume? Certainly we can discuss that. I did not emphasize that specifically, but besides working for our shareholder companies who are also our main clients, we also work with other clients who are interested in doing a project with us. And developing reference genomes is what we do all the time. But that would then be in the form of a paid project, because that's the nature of working with a commercial entity. I'm happy to discuss whether we can do that. But on the content, we are definitely interested, for sure, as well. yeah, you mentioned some very interesting names in terms of species that sound very familiar to us.
Yeah, so I want to supplement on that. So my lab and Bati from Universitat and Henry from Missouri also talking about sequencing more lagoons. So if Michiel and Tico are interested, we can talk more and to see which one is the best to do it. Because Ping has acquired a hi-fi sequencer in Hong Kong. So we can reduce the cost a lot. But anyway, I received another question from DI Farms. So could DI Farms unmute yourself and ask your question?
Thank you very much, Prof. Lam. Just a little intro of myself. My name is Sisipi Odingana. I'm an executive leader for DI Farms. DI Farms is a small agricultural holding company. We started a few years back. Our main target is to produce some leguminous plants and some staples. We also keep honeybees. And my apologies for my camera. I don't know, I'm so dark. I don't know, it's trying to emphasize that I'm really black. So my question particularly is being withdrawn by the terms that you came with, Van Eijk, on genotype and phenotype to build a model. As a smallholder farmer, I'm not really much equipped into all the biotechnological techniques, programmings, and all in depth on how to do these things. But having to work with University of the Western Cape as a research assistant for their crop research programs, it brings a question to me that the person of your caliber with your company, with the companies you work with, do you have smallholder farmers there whom probably can team up with us on this side of the world in South Africa, where we can strengthen each other in order to execute more of the sequencing you guys are talking about? We don't really understand, because from the perspective of farming, especially from the smallholder farming, we just want to produce food, especially nutritional food to the people. Well, if you can come up with data collecting programs of genotypes and phenotypes and bring models, we can assist on the ground level. For an example, currently we're working on soybean with the University of Western Cape. We're planting soybean for the University of Western Cape on the coastal area and also on the inland of the province here in Eastern Cape. So they get data that we collected in terms of, well, you can probably say in gene variation of soybean from the coastal perspective and also from the inland perspective. So I would really love to know that, do you have people that team up with us? I'm also thinking of financial constraint that we face, because as data collectors for research purposes, we really face a lot of challenges in terms of tools, equipment to bring the sufficient data that is needed. And I also had to talk about in terms of structuring, where you build other partners for shareholder values to meet those levels as well, as long as we can be able to be assisted to where we can be, then we can take from there. Thank you very much.
Yes, thank you very much for your question and providing some context. I think with respect to our own capabilities to work with smallholder farms, I think that's rather limited, also given the size of the company that we are. But I'm certainly willing to see what we can do, because what I know for a fact is that our shareholder companies, particularly I think East-West Seeds in this context, they have clients all over the world, and they have actually programs to, I think, we work with smallholder farms. So I'm happy to bring you in touch with people there. And maybe on a practical side, one of our team leaders is from here in Wageningen. So one of my 32 colleagues is from South Africa. So that may provide another, let's say, link to make a step in the right direction, so to say. But I think our own means to work with you would be very limited, because we are a contract research organization, and essentially, you know, our projects need to be paid by somebody. So I think in this case it makes more sense to bring you in touch with the seed companies who have local or regional, let's say, activities that may be of interest to you. Would that be useful?
Yeah, that would be useful. Thank you very much.
Okay.
Great. So so
Go ahead.
continue. Go ahead. I don't want to stop you. Continue, please.
No, I just wanted to say, well, maybe via Ming, you can get my email address or something. Feel free to reach out to me, and we take it up from there.
Yeah, sure.
All right, thank you.
Yeah, that would be great. So send email to us, and I will forward the Hill's email to you. Okay. So I received another question from Dr. Joanna Chan. So Joanna, could you ask the question?
Oh, hi, Michiel. It's a very interesting talk and very informative to know what you're doing. And I think it's also very specialized. So you do mention that you have many collaborators, but do you also have many competitors? Okay, that's question number one. Question number two is a very practical question, because you're working on different projects. What is the cost estimation? Just a ballpark. You can give a very small project to very big project, just estimation, because as an outsider, we have no idea what we are looking at.
Yeah, okay. Well, maybe to start with the last one first in this instance, because that's a very easy one to indeed provide a ballpark figure for, let's say, what I call the applied projects and the bilateral projects that we do with one company, because the multi-year R&D, it's a different story. But to give you a ballpark figure, a project, I think Genetwister costs between 50,000 and 200,000 euros, depending on, of course, the content. Yeah.
Then go back to the first question, right? Competitor.
This question indeed, do we have many collaborators and many competitors? Well, we are many, I would not say. We have, let's say, around 10 collaborators, so university groups or companies or consultants with whom we collaborate on specific aspects of our research projects. So yeah, it's not that many, but we're also not a big company, but it's an active part of what we do. Of course, we have competitors too. You know, Ming mentioned it. I in my previous job, I worked in a company with a very similar scope, but for different shareholders. So in that sense, you could view that as competitors, although we do not really see it that way. But I think, of course, the area of expertise where we work in is very broad and very common. You know, we are not the only one doing next generation sequencing and genotyping and genomic selection. So in that sense, there is competition everywhere. But yeah, we really do not view it that way, because the opportunity is much bigger than the competition. So, you know, we are not competing. We have our own set of shareholders, and of course they do all the work in this expertise area with us, because we are their daughter company. So in that sense, we are also a bit, let's say, shielded from competition, because we have this very stable basis to run our company on.
Okay.
Thank you.
Yeah. So all right. So I myself have a question about the phenotyping, because it could be quite expensive to install so many equipments. But nowadays, they are talking about using like a high spectral camera hooked up to a drone, so that they can collect informations, and then in the long run, it could be much cheaper than doing all this kind of chemical analysis. So what do you think about this potential? I mean, what's the developments in Europe? I mean, are they really using this kind of high spectral analysis?
Yeah, Yeah, indeed, I think it's a very important area that's also rapidly developing. I did not mention it that much, not because it's not important, but it's not. We have deliberately chosen as Genetwister not to be doing that ourselves. We think that better fits with the breeding companies themselves. You know, of course, the phenotyping itself is, of course, also core expertise for a breeding company. And this is kind of the next generation of phenotyping, again, very important. And so we know about all the capabilities of the hyperspectral measurements and the drones, you know, that are being flown. And but our viewpoint here is that phenotypic data itself is generated by the seed companies. And as Genetwister, we focus on using that data and developing innovative ways to work with that data in order to improve the traits, but not to collect the data themselves, because we do not have the scale and the financial means, and also not the, let's say, the plants themselves, you know, the breeding trials to actually collect them. We are in an office building essentially, and with only limited capabilities to grow plants, so that would be beyond our own capabilities.
So Satya, you have a question, right?
Hello. Yeah. Can I use my?
Yeah, go ahead.
Yeah. Hi, Michael. It's wonderful presentation. Congratulations, first of all. And I have a couple of questions for you. The first one is, so what is the minimum size of the population we required for Jivas? And yeah, the second one is, I have been used 3 different tools for Jivas. The first one is Tassel, and the second one is Gopit, and third one is Farm CPU. But when I compared all 3 tools results, so there is much differences. So just I am wondering about that, which tools are best to do the Jivas. And the third question is, actually I have you know that phenotypic data is like high accuracy phenotypic data is very important to identify the marker trait association in the Jivas, right? So this is my experience when I generated data from the fields. So in 2 different seasons, I have found many errors in the phenotypic data. So can you suggest me that what is like how we can minimize these phenotypic data errors to use the Jivas?
Cool. Yeah, I'm afraid I'm not capable of answering all your questions because I'm not that familiar with that level of technical detail. But with respect to the first one, I think I can give you an indication. Of course, there is no fixed number. That is, you know, the minimum size of samples for GWAS analysis, but that discussion does sound familiar to me because that's, of course, subject to debate here as well, as it also impacts, of course, on the cost of projects that we offer. So the numbers that float here is between 200 and 500 at the minimum, but in between 200 it's not worthwhile because you lack simply the power to find meaningful associations. So yeah, that at least is a ballpark indication. And with respect to the 3 tools that you mentioned, I'm afraid I do not know the answer, but I'm happy to collect the question on an email and then consult with one of our experts here and share some experience with you. Yeah.
Oh, thank you, sir. Thank you, Michiel.
Thank you so much.
Yeah. So I guess due to the time limits, we have to do our conclusion by showing some advertisement. First, of course, we will thank Michiel for all the wonderful discussions and presentation. So our coming talk will be on a different topic, right? So could Joanna put up the poster? All right, so the next talk is more on the culture. So the soybean in Chinese food culture by anthropologist Professor Angela Leung from University of Hong Kong. So we have been working on soybean for a long time, but how does it connect to Asian culture? So it could be a very interesting talk. Well, much less scientific components, but more cultural components. So if you're interested, you can scan the QR code to register, and we'll also email the registration details later. So I hope that I will see all of you again in our next talk. All right. So with that, I thanks everybody for staying so long, and thanks all the efforts by Matthew. Thank you very much.
Bye-bye.
See you. Bye.
Bye-bye.